to speak at our annual meeting today, a better example of this innovation and collaboration amongst different fields of science. I'd like to welcome Dr. Andrea Armani to Kentucky Academy of Science. Professor Armani received her BA in physics from the University of Chicago and her PhD in applied physics with a minor in biology from Caltech. She joined the Chemical Engineering and Materials Science Department in the Viterbi School of Engineering in 2008 and held the Flora Early Chair in Engineering from 2010 to 2017. Professor Armani is currently the Vice Dean of New Initiatives in the Viterbi School of Engineering and the Ray Armani Professor and Chair in Chemical Engineering and Materials Science with courtesy appointments in Electrical Engineering, Mechanical Engineering, Biomedical Engineering, and Chemistry at the University of Southern California. So Dr. Armani truly has a little something for everyone today. Thank you, Dr. Armani, for speaking to us and inspiring a new generation of innovative, collaborative Kentucky scientists.
So, assuming this goes, yes, I am gonna be a comedian. It's a little podium. I will fall off. So I first thank you for coming. I realized it was a semi-short walk, but it was a long walk for me in the rain. So I really appreciate everybody who made it. So I would like to start with this picture. So this was drawn by one of my PhD students. So she's getting her PhD, well, she got her PhD in chemical engineering, but she actually started off her academic career in undergrad as a studio art major. So this kind of her path kind of exemplifies how you can start off in one field and then completely switch into a different field. And even after she got her PhD, she was inspired by her PhD research, which was on therapeutic development, to continue and then go get an MD. So for the undergrads in the audience who are thinking, oh my God, 4 years, this is taking forever. This particular student who began life as an artist, and I'd like to point out, she's actually a pretty good one, and this is like nothing. This was a doodle, which I decided I really liked and now use it as my cover slide. She then spent 5 years getting her PhD, and then 4 years getting her MD, and she then did an oncology residency, and she's now a surgical oncologist. So she's amazing, right? And part of the reason why I think she's such a good doctor is because she's able to think about problems creatively. In other words, it's that artistic side of her brain coupled with the engineering and science side of her brain that makes her so innovative. So when you're, hunkered down in the lab, don't forget to look outside of the lab and do other things. You shouldn't just be a scientist. You need to also be an artist and a musician and a runner and whatever you love. You need to keep nurturing that side of yourself. Otherwise, you can get lost and overly focused. So that's my message to all of you. If you don't take anything else away from this talk, at least remember the first 2 minutes. I know how students are. You will tune out at this point. Okay, so about me. Julie did a great job starting with my undergrad. I'm actually from Memphis. Yes, I no longer have a Southern accent. I used to have a really strong one. When I went to college, it got beaten out of me. But I mentioned that I'm from Memphis. I went to a high school that was a really bad high school. I didn't even take calculus before I graduated from high school. This meant that transition to college was incredibly rough. Because I went to a university where it was kind of assumed that you have calculus. So day one, honors physics, everybody else in the room had calculus, and I had not. And so it was a significant step function. So it's important when you're in a classroom, you look around, just because you may not have had everything that everybody else has, as students, you need to lean on each other, right? Work in teams. I heard a bunch of talks from a bunch of amazing engineers this morning, and it was wonderful to see all of them working in teams. working together, right? Because together you're a lot stronger. So then I went to University of Chicago in physics, which, by the way, it's a great physics school. The first day of my sophomore year, one of the professors said, the only way to be a good physicist is to work alone in a library, studying problems and solving problems by yourself. That is completely not true. And so I would like to emphasize to all of you, if you ever have a professor that says the only way to do something is to do it alone in a library by yourself, just stop listening. Because the only way to move forward and move far is to move as a team. Right? So there's a saying, if you want to go somewhere fast, go alone. If you want to go somewhere far, go together. So if you want to go somewhere far in life, you need to find a good team so that you can go together. So that's the most important message. So after my undergrad, I went to Caltech. So this seems like a really smooth transition, right? I got my undergrad and I went to Caltech for grad school. Before my senior year as an undergrad, I was thinking about maybe going into finance. About half of my undergrad class went into finance. It was 2000, business was booming that summer. Everybody was going into hedge funds. That's what physicists did. They all went into hedge funds, made a ton of money. So I was assuming, I was going to do that. And then the PhD student mentor I was working with sat me down in June and said, so, Andrea, where are you applying to grad school? Not, are you applying to grad school, or are you thinking about applying to grad school, or have you considered? It was, where are you applying, and where is your list? Period. Hard stop. So just because you haven't thought about applying to grad school doesn't mean you shouldn't. And the other important message, which I didn't even know, is that if you get a PhD at a top university, you get paid. One of my main reasons for not applying to grad school, or assuming I shouldn't, is because I couldn't even fathom taking out another $200,000, $300,000 in student loans. That was something I couldn't even consider. But if you go to a top school, they not only pay for your tuition, they also pay you a stipend. I'm not gonna lie, you're not gonna get rich on it. It is a living wage. You will pay rent and eat a lot of pasta, and cold pizza too. But you can live and you get a PhD and it's great, especially if you have a good advisor. So he and I sat down, and I was actually talking with Amanda earlier. This is where networking is really important. So if I hadn't had an amazing grad student mentor when I was an undergrad, I probably would be on Wall Street right now doing hedge fund work and probably completely miserable, just based on all my friends who went down that path. A couple of them even became lawyers. That's a secondary side note. So then I went to Caltech, started doing physics, realized I liked physics, but I really liked biology more. So then I started looking into taking biology classes, which I never thought I would do. Who in here really likes taking classes? Who in here doesn't like taking classes? Oh, you're all lying. So I hated taking classes, which makes it really funny that I'm now a professor and I have to teach classes because I'm teaching something I don't like. I'm not to brag, I'm good at it. I get 5 out of 5s on my teaching exam. But I really disliked taking classes. So when I took my last class as an applied physics PhD student, I went out and partied. Because I was I never have to take an exam again, this is awesome. And then the next quarter, I was I think I'm gonna take a biology class, which means I'm gonna have more exams, and I really wanna learn biology. So I got to take really cool classes because I wanted to. And that's a very different attitude than taking a class because you have to. So I had a lot of fun, and then I did a postdoc in biology and chemical engineering. So these are two very different fields. So my applied physics PhD was in a field called quantum optics, which is really just studying how photons interact with each other. And my postdoc was in developmental biology, studying how this really cool little fish that's transparent, that's called a zebrafish, how its heart develops. So totally different, but very fun. And then I went and started applying for faculty positions, which, to be honest, is like a lottery. And kind of like a lottery where you're blindfolded and throwing darts at the lottery tickets. So I applied everywhere and to a lot of different departments. Many departments I had no degrees in, just crossing my fingers and hoping. And I was really lucky that I got a job at USC in LA, which is where I was already living. But that was luck. If you notice, I'm in a chemical engineering and material science department, and I have no degrees in chemical engineering or material science, but physics is kind of like material science, so it works out in the end. But I'd also like to point that out for the students who sometimes are like, yeah, my professor seems like they don't know what they're talking about. They might have a degree in mechanical engineering, and they're teaching you material science, so they're kind of learning it along with you. So be nice to them. But we do care, we really care about you, and we're trying our best. Okay, so my undergrad. The first research project I did, which is kind of like an academic project, was on something called diblock copolymers, which now has been, people have been working on it for like 35 years because I'm old. I'm gonna lay that out there. So diblock copolymers were really, really cool at the time because you can take this little tiny polymer structure, but you have two of them. You have like an A part and a B part, and when you spin coat it, you get this pattern that looks kind of like a zebra stripe, and then you can deposit metal on them, and the metal only goes to one of the polymers, so then you get little tiny stripes of metal. And for all the electrical engineers, you can imagine now you can make little tiny self-assembled circuits. Or at least you can make little tiny self-assembled wires. So that was the vision. That's still the vision, like 30 years later. People are still trying to do that. My project was just trying to make the diblock copolymers have straight lines, because they were having problems making them have straight lines. But this was really my first introduction into the field of chemistry and physics, how chemistry and physics overlap. Because in order to understand why we weren't getting straight lines, we had to understand the physics of the interaction between the polymers and the substrate. But then in order to control the width of the lines, we had to understand the chemical structure of the polymers. So it really required both pieces of information. And I went to the organic chemist that was actually making our polymers, and he told me he wouldn't tell me anything about the polymer structure until I'd taken organic chemistry. And I thought this was very unfair because I was already taking all the physics classes. But I went ahead and took the organic chemistry class, and I'm really glad he made me do it, because if he just told me the structure, I now understand why. I would have not understood anything about what the structure was telling me. So if you're thinking about taking a class that is completely out of your field, you should do it, because you never know when it will come in handy. So I worked with a person named Heinrich Geiger and Ward Lopes for a couple of years, and then for my senior thesis, I did something totally different. I went and started working with lasers. And I worked with optical tweezers, which, who knows why optical tweezers and optical traps are notable? Anybody? It has. There have been two really big prizes given for them. Bingo! Excellent. See, this is interactive. Okay. Yes, so the Nobel Prize was given for the optical trap. So I was developing, instead of a single point trap, I was developing a line trap and then studying how things could move along the line. But this was my first experience using lasers. And, pew pew, You can put a laser on top of a shark's head. So I was really having a lot of fun using high power lasers, learning how to align lasers, learning about laser matter interaction. And around the same time, people started looking at using lasers to measure biological things, like the Young's modulus of DNA, or the binding energy of individual base pairs in DNA. You know, how much energy did it take to like rip DNA apart? And I decided I wanted to do that. I wanted to do those measurements. I wanted to study the energetics of proteins and how they fold and unfold. And this was one of the only ways to do it, was using optical traps. And since I understood the optical trap part, now maybe I could start doing the bio part. So I started looking around for different potential grad school places with, you know, my grad advisor, my grad student advisor, to try to be able to do that type of research. But in parallel, right, like that's the sciencey half of me, I also did non-sciencey stuff. So I was in like student clubs, like the physics society, the chemistry society, and I played soccer. Soccer was great for me as an undergrad. It gave me an entire group of friends that were not scientists. It gave me people to go hang out with on Friday night. So make sure you have that non-science part of you. And you have friends that you can just do other things with. It's really important. Because then you can also complain to them about, you know, other things. You know, you use them as like a sounding board. So then I started looking at grad schools. And I ended up signing up to work with a person named John Crocker at Caltech. He was like a second-year assistant professor, super young, very energetic. He was doing exactly what I wanted to do. But day one at Caltech, he pulled me into his office and said, Andrea, you know I'm married. I have a wife. She's an art history professor. She's at UPenn. I was able to get a job at UPenn. A lot of the faculty in the audience know where this is going. So I'm moving to UPenn. You are welcome to come with me if you want to, but I also understand if you want to stay. I wanted to stay. I had just come from Chicago, where it snows. I had no interest in going to UPenn, where it also snows. I was really excited about being in Los Angeles, where it doesn't snow. So he offered to continue to pay me for like the remainder of the year until I could find somebody else to work with, which was very nice of him. And so I started looking around. So my second PhD advisor is someone named Harry Atwater. Love Harry, great guy. I mean, by the way, I love John too. He's a great guy, super nice. Harry Atwater, awesome. Harry does plasmonics, which is this field that studies how electrons can hop between gold nanoparticles at like the nanoscale. That's a gross oversimplification of the field, but let's go with it. So he was going to let me make plasmonic biosensors. So I was still going to get to do bio stuff and light stuff, right? So I was still going to get to do what I wanted to do in a slightly different research group. Everything was fine. I actually only had to move my office like up one floor in the building, which was really convenient because I had a lot of books. So this was like, I don't know, October of my first year in grad school. January of that same year, Harry pulls us all into a conference room and says, hey guys, we're moving to Harvard. If you're a second year grad student and above, everything's cool, don't worry, all your courses will transfer, you'll get your PhD at Harvard, everything's great. If you're a first or second year, you're gonna have to take all your courses again. And you're gonna have to take your screening exam again. So, you may or may not wanna continue taking your courses. That doesn't sound like a lot of fun. So I started looking around for another advisor. Just as a side note, 2 years later, Harry moved back to Caltech. Not okay. I'm going to continue this story. Okay, so I started looking around for another advisor, and I found another advisor who will not be named for reasons, but he was doing single molecule microscopy. Really fun, something called TIRF, or Total Internal Reflection Fluorescence Microscopy. He again was a new professor, he was in his third year. So this was at a time when you couldn't buy a TIRF microscope, so you had to build them from scratch, which was really fun because I really like building stuff. So I got to use a dye laser, couple it into a bunch of free space optics, run it up the back of a microscope, set up the evanescent wave, take single molecule images. I worked with a postdoc who was a really good programmer who developed the software to actually capture the images. We then did some microfluidics in order to improve the resolution and the throughput to study individual binding kinetics between proteins and whatever they're trying to attach. So super fun research. The reason why he was a chemist, he was in the chemistry department. The reason why he was really excited to get me into his group is because he'd been there 3 years and he hadn't been able to get any of the chemistry PhD students to be able to build the TIRF microscope, which was at the cornerstone of his research program. And this should have been a warning bell in my mind that he'd been there 3 years and didn't have the piece of equipment he needed to do his research. And in June of that year, he didn't get tenure because he didn't pass his mid-year review, which is why I'm not naming him. So at this point, I'm heading towards my 4th advisor in under a year, which, just as a side note for all the undergrads, this is not a thing at all. Usually a PhD student might change advisors once, maybe, usually 0 times. 4 is just, you just don't. That's not something that happens. To the point that when students would think about changing advisors, the Caltech student affairs person sent them to me to counsel them about options and strategies. It was bad. So then I started looking around for my 4th advisor. And at this point, I didn't care what I did. I was like, I will do theory, I will do experiments, I will do protein crystallography, just sign me up. I just want an advisor, I just want out. My new requirements had nothing to do with the topic. They had to do with, I want a PhD advisor that's not gonna leave, critical. I want one that's tenured and chaired, and that's all I care about. That was my new search criteria. So Kerry got his undergrad, PhD, postdoc, and then took a faculty position at Caltech. His house was 3 doors down from Caltech. Seemed like a pretty solid anchor. Wasn't going anywhere. And then his research was on quantum optics, so it was still light. No bio, still light. And he had just started a startup company, and he was on a 3-year sabbatical. So my deal with Kerry was that if I brought in a grant to pay for myself, he would just let me do whatever I wanted. That was my deal. It was a great deal. It worked out. I would not recommend that deal to anyone. It was a bad idea, but it worked. So the big message is that, when you have a plan, when I went into grad school, this was not my idea. Right, I had this really nice, clear-cut plan of exactly what I was gonna do. Everything was gonna be perfect. I was gonna work with John, who I had a really solid working relationship with, and then everything just kind of detonated. But in the end, I got my PhD, shockingly enough, in 5 years. Still don't know how that worked out. And I ended up getting a solid postdoc. So you can navigate. And I don't know, 10 years later, I managed to wiggle my way back into doing biooptics. It just took a little bit of a time. And while I was there, and again, this is probably what kept me a little bit grounded through all of that journey, I was in the Graduate Student Council, and I was also part of the Women Mentoring Women program, which was really useful, because again, that gave me kind of a nice group of people I could talk with that were independent of everything else going on. I also had an amazing boyfriend, then fiance, then husband eventually, and currently, who's amazing. So then my postdoc began doing bio-optics again, did imaging again, did some chemistry, and then went to USC. So I've now been at USC for 14 years, and I will show the research in a little bit, but the biggest and most important part about being a faculty member are all the students. So these are pictures of all of my students over like the last 14 years, and they are, from like a faculty member perspective, like I do research, but when I think about the impact that I've had, it's all of them, right? It's not the research results, because the students are really the most important thing, because they go out into the world and they have the actual impact in the world, not really me. I'm like the secondary impact. I make impact because of them. Although 2 of them have started companies, so I'm totally claiming credit for them. We were talking about IP earlier. It's not my IP though, unfortunately. Although I will say they did promise if they sell their companies for more than $100 million, they're gonna give me 10. So I'm really excited about that. They aren't there yet though, but I'm already looking at the island I'm gonna buy. I'm joking. Okay, so this is my current research group. I split them all into their bins of their degree programs, and I always like to do that when I'm giving a talk like this, just so you can see exactly how interdisciplinary it is, right? When you see kind of my research group as like a whole mass of people, it seems really gigantic, but actually it's not, right? Because there's, a couple biomedical engineers, a couple material scientists, a couple chemists, a couple electrical engineers. So it's actually this really nice puzzle where you only have a couple of each field and they all work together. So when there's any given project, and maybe there's like an electrical engineer, a chemist, and a biomedical engineer, like working on kind of a little tiny project together. So it's not 15 biomedical engineers, right? There's a lot of people from a lot of different backgrounds, and they all learn from each other. And that's really important when you go into the project and you have your own expertise, but to learn from everybody around you, and ideally to be in an environment where everybody around you has different skills than you do, so that you can constantly be learning from them. And then I have 9 undergrad students right now. So I have almost as many undergrads as I do PhD students and postdocs. I love undergrads. They're great. I also have 1 high school student. High school student takes a lot of time. It's super adorable. So I'm often asked, why do I have so many undergrads and a high school student? So as you get older and older and older, you can become more jaded and set in your ways on doing research. And part of this is because if you do something one way, it'll work. And so the idea of doing something a slightly different way can seem a little risky, and you'd rather just do it the way will work. So this kind of becomes evident in doing things like, I'm going to use an example from this morning, programming. So MATLAB has been around forever. It's a great programming language. The 2 students in the back know who I'm talking about. But there are so many other programming languages, and one of them is Python, and it's new. And most faculty have never heard of Python unless they're actually in CS, but it's free. And free programming languages, which are open source, are awesome. But again, unless either you went to college in like the last 4 years or are a high school student, shocking, then you probably aren't fluent in Python. So like my high school student comes in sideways because he's never done MATLAB. He has no access to MATLAB because he's in high school. And he starts working with my undergrads, And my undergrads are, like they're using MATLAB because that's what they learn in class. And my high school student has the option of doing something in Python, which he knows, or trying to learn MATLAB, which he doesn't know. And his option is, he's like, yeah, why don't we just do this in Python? It's free. There's this whole community out there that knows how to do this. It's a lot easier. We can then just post it all on GitHub. I'm like, yeah, we should do that. That sounds really good. Thank you for that. Do you have any other ideas about how you can make my research better? You cute, adorable 14-year-old. So that's why high school students and undergrads often are great, because they come in completely untarnished by the rigor that is the academic institution. So for all of the undergrads, that is your strength. The fact that you actually haven't been trained, right? The fact you don't know as much, so you think completely out of the box. So don't worry about proposing ideas that are opposite of what your faculty may say, because they actually might be so much better. So that's a very important lesson. And that was like an actual example that my high school student did. Like, that wasn't hypothetical. And now we use Python for everything in my lab. And it's great. And it's free, and I don't have to pay MATLAB anymore. It saves me so much money. Okay, so in my lab, just the really fast research evolution, we started off making lasers and then continued making on-chip devices, optical devices. So optical devices are like electrical devices, but they conduct photons instead of electrons. So that's the easy way to think about them. Then we started making materials. And by materials, I mean, we started making nanoparticles, like gold nanoparticles, quantum dots. We then branched into polymers. So we went from inorganic synthesis to organic synthesis. We did some on-chip reactions. So we actually grew the polymers off of the wafer. I like to think of this as like growing grass off of our wafers so that we could get vertically aligned polymers as opposed to horizontally aligned polymers, and this helped make better optical devices. So it was a whole like feedback loop. This wasn't like a shotgun approach to doing research. There was a vision. And then a lot of that work fed back into making better diagnostics. And then again, my personal goal of making diagnostics and making materials kind of began to grow farther into not just making like a diagnostic chip, but actually making a system. And making a system is the unappreciated part of science. It's really easy to make like a single chip that works in isolation. It's really hard to make a system. For example, making a solar cell system that you can stick outside. It's really easy to make a solar cell system that works in, nice ambient environments, but making one that you can stick outside and works in the rain and the snow is really hard because you have to address a lot of issues. So that's what we started working on, was making diagnostic systems that were lightweight, had long battery lives, could be rechargeable, that could be fixed easily, so you could actually get to the inside and replace components. So we started working on things like that. So now our group does a little bit of a lot of things, which is why we have a handful of people from a lot of fields. So we do some materials work, some device work, and some systems work. And we work really in kind of two different areas. So we do some integrated photonics work and some what is now deemed health and wellness work. That's like the new fancy way of thinking about doing diagnostics and like disease work. So it's, you kind of like flip your mindset. Instead of trying to like cure diseases, you try to keep everybody healthy. But it's, let's be honest, it's the same thing. So it really combines kind of the chemists and the optics and physicists and the biology that I worked on developing for a very long time, but it combines it all into like one research group, which is super fun. But today I'm going to give a brief example of one of the research projects in the group, but obviously there's a bunch. I'm going to try to do this at both a high level and a specific level. So we're gonna see how well I pull this off. Okay, so bioelectric fields. So bioelectric fields are one way that cells communicate with each other. So there are lots of ways that your cells talk with each other. Right? So usually when you think of cells talking with each other, it's through chemical ways, right? So like maybe antibodies talking with each other, maybe chemical gradients like pH gradients, salt gradients. And then with neurons, most people also think about electrical signaling, but actually almost all cells talk to each other with bioelectric fields. It's very common in wound healing, in fact. So if anyone has ever had a surgery, like an orthoscopic surgery with like a very large open wound, and I mean recently, in like the last 5 years, they might have given you little tiny external like stickers that you could put on it, and then it would put in RF energy that would stimulate the intrinsic bioelectric fields in your skin to try to increase wound healing. So that's improving endothelial bioelectric fields to try to improve healing. So if you think about cells as being like a wireless network, because they all talk to each other in lots of different ways, and this is going back to kind of all the EEs in the audience, if you think about it as being a wireless communication network, then when the network is all happy and strong and healthy, all of the different cells should be able to communicate with each other, right? So that's your happy, healthy tissue. However, if there is one cell that dies or is dying or drops out of the network, then suddenly your network is weaker, right? And there are lots of different questions that computer scientists and electrical engineers often ask when they're trying to model this type of system, right? So, how robust is it? What is the minimum number of nodes that you need to have in a wireless network to maintain connectivity? How fast is your network? If your one node drops, how fast can all the other nodes kind of kick back into gear to restabilize the network? And if any of you have ever been in a building where it has unstable Wi-Fi network, it's because of nodes coming in and out and the inability for the entire network to restabilize due to increasing and decreasing activity at one of the nodes. So this type of modeling is done by a lot of computer scientists. So you can do the same type of modeling for biological networks. And actually, when non-biomedical engineers realized that they could apply some of the same equations to biological systems, it opened up a huge field of biology, trying to apply systems engineering to biological systems. So using simple circuit designs for cell signaling, for example. So in this case, these are neurons, right? So if you start thinking about how neurons are connected, how is information transferred between neurons, if we cut out one of the neurons in the middle, then how does that impact all of the other neurons being connected? Now, really practical question, why should we care? Well, if you think about neurons in the brain, right, like what happens if one neuron is compromised, right? Like how does that impact how all the other neurons around it function? Probably not much. But what happens if 2 or 3 or 4 or 5 neurons are compromised, right? Then you're going to suddenly have significant decrease in your neuronal network connectivity, right? And then what happens if they're kind of compromised, but not really? Like how fast can we recover them? So that's why this is an interesting question. And again, it's not just neurons, right? A lot of heart cells are controlled by this, so myocardial heart cells. And obviously all of you know this because when you have a heart problem, right, you get zapped with the electrodes. But there are lots of characteristics of interest, like spatial extent of connectivity, temporal aspects. So in order to really understand this and ask questions about this, we not only need to be able to precisely control where the cells are located, we need to also be able to turn them on and off in a test system. So there are lots of different, oh, that changed. So there are lots of different ways to actually monitor if a neuron is on or off, right? And you've probably seen pictures that look kind of like the one, like this one before, where the neuron cell body is all bright and green, indicating that a neuron's actually there. And then you have the, I'm trying to think of a good way to say this, the region on the neuron where the pulse travels, and then the ends, which will then signal to the next cell body at the end. And this, complexity makes it very hard to actually watch neuron firing, simply because this is so complex, right? There are so many neurons there, watching them turn on and off is very difficult. Additionally, with this system, you can see if a neuron is turning on and off, so you can get that reading capability, but you can't necessarily make it turn on and off, at least not quickly. So there are a few ways of actually doing reading and writing, so basically monitoring if a neuron is firing, as well as make it fire. These are kind of 4 common ones. So the first is an electric probe. So you literally just shove an electric probe into a tissue sample. That is as damaging as it sounds. The second is doing a patch clamp. So you patch, you suck a little part of a cell into a cuvette and then control it. And the third is a super fancy way called optogenetics. So optogenetics, you take a tissue section, you do genetic modification, and then you're able to control a lot of different things about a cell or tissue section. And optogenetics is super famous now, and people are hoping that you could genetically modify a lot of different systems and then control them with blue light. One challenge about that is that doing genetic modification intrinsically changes the thing you're studying. And one of the kind of fundamental rules of science is if you want to study something, you don't want to change it, because if you change it, then the thing you're studying isn't the thing that you actually want to study, right? You want to study a thing, not a variant of the thing. So a read-write molecule would be able to both detect the field as well as modify the field in one go. So that was our vision 5 years ago. So I want to emphasize that we've been working on this for 5 years. So this is going to seem super easy, and it wasn't. So we wanted to have something that would insert into the cell membrane and then be able to simultaneously control the bioelectric field or the electric field across the membrane, as well as read out or tell what the electric field across the membrane was. So we designed this molecule that had 2 sides to it. So you can think of it as a molecular device, right? Because it had one arm that had one function, another arm that had a second function, and both arms could be optically controlled. So you didn't need to have any kind of probe physically in contact with the sample. And this concept came out of the idea of having a fluorescent probe that you didn't have to physically couple to, you could optically couple to. So briefly, the 2 parts, one is a modulator. So all the EEs in the group should know all about modulators, because they're a fundamental component of a lot of circuits. So this is a photoconductor, which again, solar cells, yay! So we basically stole the actual functional group from organic solar cells. So we used the active group of organic solar cells, which means we put light in, it spits electrons out, so then we're able to inject electrons into our cell. We then used a reporter as the way to sense the electric field around the cell. So it's a 2-photon reporter. And for the chemists who are super excited and want to see the molecular structure, so the molecular structure is a combination of a TPE reporter and an NAI modulator. So we did molecular design. And again, this is really for the chemists because I know there are a handful of those and I know they're going to ask me. So we did DFT modeling in order to optimize the absorption and emission wavelengths of both our modulator and reporter, because one concern is obviously that the modulator and reporter are going to interfere with each other. In other words, we're going to get crosstalk, right? And you don't want them to crosstalk with each other. So we did DFT to optimize that. And then one point, which is also important, is this little tiny carbon chain here. So that carbon chain acts to electrically isolate the two functional elements from each other, so that you don't end up with electron transfer between those two groups, which would then just nullify the entire structure. So we also had a little spacer molecule. And for the synthetic organic chemists in the audience, this is how we made the sucker. So that's the whole reaction. If you're really excited about it, we can talk after, because there's probably going to be like 2 people who care. That's my ballpark. So we have a bunch of graphs, but really, I always say science is glowy materials. So it worked. Victory. The most important unique property about this material is that usually when you have a light emitting material and it's an organic dye that's light emitting, when you increase the concentration of it, the emission intensity goes down, because you end up with the dye molecules transferring electrons between each other, and then you don't have as much emission. This material is particularly unique. It undergoes a behavior called aggregation-induced emission, which means that as you have more of it, the emission actually increases. This is because the molecule actually has internal molecular rotation, and as you inhibit that rotation, you get more energy that is emitted, which is super cool, because there aren't that many molecules that do this. So this is an example. This is low concentration of the dye, and this is high concentration of the dye. And you get a lot more emission. This is important because it means that when we take our dye and we put it in the cell membrane, where that molecular rotation is inhibited, it will light up. But any dye that's not in the membrane won't appear. So it means we're actually going to get enhanced performance with the dye that's in the membrane. But if a dye isn't in the membrane, it's not going to light up. So then our background will be really low, which is a victory, like really big victory for imaging. Then we also check the photoconductivity. So this is kind of a classic photoconductivity measurement. We in my lab, we did not do this. We don't have electrical characterization equipment. We worked with an EE lab. So we looked at the dark current, we looked at how fast it can respond, we looked at how much energy. Most important thing is it's still a photoconductor, even though it has that fluorescent tag on there. We were kind of worried. However, we have to be able to actually stimulate it, right? Not just with a high energy beam. So we need to be able to precisely control and turn it on and off when it's in tissue. So we wanted to do that using optical fiber. So we're going to start off with just conventional classic optical fiber, like the kind that you use for internet. And then we're going to strip off the cladding, and then we're going to use a hydrogen flame to pull it. And I mean, like, we have a gigantic hydrogen tank in our lab with open fire. And then dip it in BOE, which is basically hydrofluoric acid with a little bit of buffer in there, and sharpen it to a point. So we start off with kind of this blunt end, and then after it's tapered to a point, we change the profile of light coming out the end so that it's much sharper and much more confined. And it's predictable. We did modeling also to predict that. Okay, so conclusions, and I think I still have time for questions. I really hope so. Excellent. So we made this molecule, and it works in cells, and it doesn't kill cells, so that's a victory, which like everything kills them. And then also we made the way to actually trigger it, so we're now working on that. It also, I didn't show the data, it also doesn't kill neurons, which is a big victory. And again, my students do everything, so I always like to end a talk by thanking them. And I'm very happy to take questions, especially from all the engineering students that I asked questions of this morning. It's their time to get back at me. Thank you. There are microphones here if you want to have a microphone, or you can just talk. Questions?
Engineering students want to get back at her? Really hard questions? All right. Oh, sorry. So you said the molecule you have is activated in the membrane of the cell, right?
Mm-hmm.
Why? How? What's in the membrane that's not in other things?
Yep. So it is active in the membrane because of how. So it's active outside of the membrane too. It's just here. I'll go back. So if you look here, right? So both of these have the same amount of the molecule in the solution, right? They both have the same amount of solvent in the bottle, but they have different concentrations of the molecule in the solution. And you can see a little bit of fluorescence in this flask, but it's really, for the most part, just reflecting the fluorescence from this flask. So outside of the cell membrane, you will have some emission, but it will be very, very, very low. When the molecule is in the membrane, because the molecular rotation of the molecule is inhibited, because it's in a fixed area, because the bilayer of the membrane acts basically as having a higher concentration of the molecule, then the emission intensity will increase because of this aggregation-induced emission behavior. So the molecule doesn't have to be next to other molecules of its type. It just has to have that motion inhibited. So the same behavior is seen if you take the molecule and put it in like PDMS, like a polymer matrix. You see the same behavior. So it just has to not be able to wiggle.
Thank you. That makes a lot more sense. But I was also wondering if by in the membrane you mean inside of the circle, kind of right, like inside of the cell, or inside of the membrane itself, like
inside of the membrane.
Okay.
Like that.
So if it's in like the cytoplasm or whatever, doesn't.
So like that's the outside of the cell membrane. So inside the lipid bilayer part of the cell membrane. Here, hold on. Like in here.
Okay, thank you.
Yeah.
Is it cell permeable then?
Is it still permeable?
Is it cell permeable?
So we know it gets into the cell membrane. We don't know if it makes it into the cell.
Is it cell permeable?
Yeah, because when it gets into the cell, it can then wiggle around again.
Exactly.
So we can't really tell.
Okay.
In the main work, we did like some work with those cancer cells, which is what the last slide was a picture of. The majority of the work we've done is actually with neurons, and if we try to hit them too hard with light, then they just get really unhappy and die. Like everything we do, yeah. Yeah, makes sense, yeah. And we've actually now started, I actually removed this slide. We now have a way to bioconjugate or to add an NHS ester group onto the end, so then we can do bioconjugation, so then we can selectively target specific things inside the neurons, inside the membrane, inside the neurons, so then we can tell which type of neuron inside a I call them brain balls, but that's not the technical term, neural microtissues. So they basically take little tiny, I really hope I'm not about to get in a lot of trouble here, little tiny rat brains and then grind them up and then recapitulate them into neural organoids. Great, nobody's barfed yet. Okay. I was waiting for it. And then these little tiny neural microtissues is what we're working with. So they have all different types of cells. So we're working on bioconjugating them with either antibodies, which are gigantic, or peptides, which are smaller, so that we can selectively target specific types of cells within this heterogeneous matrix of neural microtissue. So we kind of skipped a step. But our collaborator who does all the cell work, that's what she does. So we skipped it. We tried to convince her to do 2D work, and she was like, yeah, I don't do that. So, but then it worked with the 3D, so, yep.
I'm one of those organic chemist people, so.
I knew.
Too. So, I'm curious about your synthesis, just looking at the structure. Did you incorporate in your thought process of the design that you wanted that aggregation, and what was your thought process on the design of the compound?
So we had a lot of thoughts on the design of the compound, many of them. So we're already actually like redesigning it. So I'm just gonna start there. So this backbone, this part, is way too oily. So we're now replacing it with a PEG to try to make it a little less oily, to make it a little bit, there's kind of a balance of making it more hydrophilic so that it gets into water around the cells a little better, but then you also want it hydrophobic so that it will actually get in the cell membrane. So there's kind of like this delicate balance, but we want it a little bit more hydrophilic than it is right now, so we're actually replacing that with a PEG backbone. So that was one consideration, was this. We went with just a simple carbon chain for the first route because the reagent was cheaper. I'm just gonna be honest about that. Then we have 2 of the NAI groups because being able to modulate, we decided was really important. And I view these as like 2 antennas. So if we have 2 of them, then our absorption cross-section is gonna be better and we'll have a higher probability of modulation. And the efficiency of the NAI isn't awesome. So that's why we put 2 on there. We tried it with 1 and it didn't work very well. So when we stuck 2 on there, we were able to get better electron generation. The TPE module, we tried a couple different lengths, and this one gave us both the highest emission intensity, but also we looked at putting different groups down here to try to redshift it a little bit, and having just this structure was able to optimize the absorption emission wavelength of the TPE with the absorption wavelength of the NAI. So that was the whole design. We're now also looking at changing this group because we're doing some work in bacteria where this needs to be more positive. So we're also looking at changing that group for the bacteria work. So it's very modular. So we're kind of changing a lot of different things. But just getting that, like we had a lot of false starts.
Hi, I'm back here, by the way. This is loud.
Yeah, I was gonna say, I hear.
Yeah, this is loud. I'm back here, by the way. First of all, I wanted to say I appreciate you for coming out. I very enjoyed your speaking. But you had said on one slide that you've done lots of impactful research, but perhaps the most impactful thing you've done is being an influencer on a lot of younger people. And so I was wondering, what advice would you have for an undergraduate researcher to be able to produce the highest quality of research at the undergraduate level?
So I would say at the undergraduate level, really, oh God, faculty are gonna hate me. You should think about your undergrad research both as trying to do something very high quality, but also as your chance to learn something that you aren't gonna learn in your classes. So I always encourage my undergrads, like if they're majoring in biomedical engineering, to do a research project that's more mechanical engineering oriented. So think of it as something, a chance to do something different. Almost like you're getting a minor in something else, right? So to really dig in deep. So one mistake I see undergraduates make is to try to do as much as possible, but then they end up doing something very superficial and they don't really understand what they did. So it's better to do something small, but really understand it, really understand why you're doing it, really understand every decision you've made, really understand the motivation of the whole project within the field, as well as the motivation and the decisions you've made in your project. So I really hope I didn't just make every faculty member here really mad at me. So I wouldn't necessarily say it's like, how do you do the highest impact research in terms of the field? It's the highest impact on yourself, right? Because your undergrad research is really an investment in yourself and your education. So that would be my advice. Also, learn how to use stuff, right? Because learning how to use a piece of equipment is a life skill that will translate for any field. So learn how to use equipment, like as much equipment as possible, especially machining. Like take a machine shop class. Like it is a dying skill set, but understanding the difference of like drilling into a piece of aluminum versus drilling into a piece of steel versus drilling into a piece of plastic really goes a long ways when you start doing a SolidWorks drawing to then design something for, I don't know, solar cells, because you will have a really good understanding of design tolerances, and you won't design something that's just not functional and impossible to machine. So my husband owns a small engineering company, and that's his biggest complaint about undergrads right out of undergrad. Is that they design things that aren't physically realizable. I believe that is his phrase, physically realizable, or they're not physically realizable. So just like, at least spend like 2 hours in a machine shop, just getting a feel for what the different materials feel like, as opposed to reading about them. Yep.
From an information transfer perspective, like that could be considered at the local level, but then you also mentioned like the network level.
Yep.
As far as the neural network and electron transfer, think about like alpha helix and the phonon characteristics of alpha helix. How are you transferring information from like one particular aspect at the lower level to like the whole area network? Have you all done research on like the phonons or the alpha helix polymer level? Have y'all done research on that? And what have y'all done?
Yeah, we have not looked at it at like an individual protein structural level. We've really been looking more at like a cell level. And that's just because that's really been where our focus is to try to develop a tool to look at like cell-cell communication and to control cell-cell communication, understanding how electrons flow within like an individual antibody with the different structure. Like that's a really interesting and complex question. We just haven't even looked at it. It hasn't been on our radar. It's a really good question. Yeah.
So I'm an animal social behavior.
Yep.
Completely different area.
Mhm.
I'm very curious about how you manage a lab. It's a large group of people.
It is.
I'm sure there are lots of conflicts that come up, but also lots of good things.
Mhm.
How do you manage it so that the good things happen and the conflicts don't cause things to fall apart?
So the question was, how do I manage my lab and keep everybody happy? I'm gonna really simplify it to keep it positive. So I have, I'll start with, I have a pretty rigorous management philosophy, which is actually very clearly stated on my group webpage. So everybody who comes in, I don't want to say has like signed up for the management philosophy, but they're on board with it. So I meet with every group member every other week. So I've actually split my research group in half. And I have the groups kind of loosely arranged by topic. But I meet with everybody for half an hour every other week. So I get individual time with everybody, which allows me to kind of keep a pulse on how every student is doing as a person, but also as a researcher. So that I can tell maybe, especially with the first year PhD students, if they're struggling a little bit with the transition from undergrad to grad school, or so I can just monitor that. Then we actually have something called the Armani Lab Fun Club, or AFC. I give them money every semester. I don't ask what they do. I know they plan a lot of different activities. The most recent one was a game night where they played Uno. They acquisitioned one of the conference rooms, watched a movie. I give them money and they plan things. They went to Six Flags one time, like the Fun Club co-sponsored the Six Flag trip. That may seem really expensive, but they actually also did a fundraiser. So I have a group party at my house 3 times a year. And I had a group party in June where I also invited all of the Southern California area alums. So then my very enterprising current students took some of the Armani Lab Fun Club as like a catalyst or seed money and had a Sunday stand and then sold Sundays to my group alums and raised like $5,000. Like, great, so my alums give my current students money. Like, why didn't I know about this as like a cycle, like a thing I could do? So that was wonderful to see, like my alums supporting my current students. So there's also that. So now I think my current students maybe still have like $2,000 left, but I just give them money. So they're a very tight group of people, and it's in part, oh yes, and also when I do admission for the PhD program every year, I down select to about 10, but I only admit about 5, like 3 to 5. And once I get to 10, I circulate the CVs and the personal statements of the top 10 to all of my current students. So all of my current students have a voice in who is admitted. So they feel like the students we admit, they're already part of the family. So it's not as though I'm admitting somebody and then foisting that person upon the team, right? The team has voted to bring that person in. So it's a different mindset. And then the group is actively involved in recruiting the person to the team. So it's a very different way of recruitment and student involvement and engagement. And of course there are hiccups. I would say the majority of the friction revolves around, what would I actually say? So the most recent was about authorship on a journal paper, but we've actually only had 3 authorship on journal paper things. We have like a spreadsheet where whenever there's an authorship issue, everybody fills out the spreadsheet. It's like a point-based system. There's like a whole thing where like points are based on, like who came up with the concept, who developed the approach, who actually did it, who actually wrote it, who analyzed the data. Like there's a whole bunch of points, and then you write down everybody's name, and then you assign points. And then usually it ends up being really clear, like, and everybody's points pretty much agree with each other, and then there's no argument anymore. So that usually works out. But yeah, we actually have a pretty good group. There are very few arguments. It's nice.
Great, well, thank you very much.
Thank you.
That might be it for us.